Today, we will be exploring the fascinating world of medical nanotechnology and its implications in antimicrobial therapy. As we delve into this cutting-edge field, we will examine the various types of nanoparticles used in antimicrobial applications and their potential in combating drug-resistant infections. With the alarming rise in antibiotic resistance, nanotechnology offers promising solutions to enhance the efficiency and effectiveness of antimicrobial treatments. Nanoparticles, defined as particles ranging from 1 to 100 nanometers in size, possess unique physicochemical properties that differ from bulk materials. These properties, such as increased surface area to volume ratio, allow for enhanced drug loading capacity and improved drug release kinetics. Nanoparticles can be tailored to carry antimicrobial agents and deliver them to specific targets, maximizing therapeutic efficacy while minimizing side effects. Metallic nanoparticles, such as silver and gold nanoparticles, have emerged as powerful tools in antimicrobial therapy. Their inherent antimicrobial properties stem from the ability to release metal ions, which exert toxic effects on microbial cells. Additionally, metallic nanoparticles can disrupt microbial membranes, interfere with cellular signaling pathways, and induce oxidative stress, ultimately leading to cell death. The unique ability of metallic nanoparticles to target specific microbial pathogens makes them promising candidates for combating drug-resistant infections. Biofilms, characterized by microbial communities embedded within a self-produced matrix, pose significant challenges in antimicrobial therapy. Polymeric nanoparticles, made from biodegradable polymers, have demonstrated potential in eradicating biofilms. These nanoparticles can penetrate the biofilm matrix, releasing encapsulated antimicrobial agents directly onto microbial cells. By disrupting the biofilm and targeting its inhabitants, polymeric nanoparticles offer a promising approach to combat biofilm-associated infections. Drug resistance is a growing concern in the field of antimicrobial therapy. Liposomal nanoparticles, composed of lipid bilayers, possess unique properties that make them ideal carriers for delivering antimicrobial agents. These nanoparticles can encapsulate hydrophobic drugs, protecting them from degradation and enhancing their stability. Moreover, the bilayer structure of liposomal nanoparticles enables them to fuse with microbial membranes, delivering their contents directly into the cytoplasm. This targeted drug delivery approach holds great promise in overcoming drug resistance in microbial pathogens. Quantum dots, semiconductor nanoparticles with excellent photophysical properties, have garnered attention in antimicrobial therapeutics. These tiny nanocrystals can emit fluorescent signals, enabling real-time imaging of microbial cells and biofilms. Moreover, quantum dots can be functionalized with antimicrobial agents and selectively delivered to microbial targets using ligands or antibodies. This dual-mode functionality, combining imaging and antimicrobial capabilities, provides a versatile platform for precise antimicrobial treatment. Nanoemulsions, composed of oil droplets dispersed in an aqueous medium, have emerged as effective carriers for topical antimicrobial applications. These nano-sized emulsions can encapsulate hydrophobic and hydrophilic antimicrobial agents, improving their solubility and stability. The small droplet size of nanoemulsions allows for enhanced penetration into the skin, ensuring efficient drug delivery to the site of infection. Nanoemulsions offer a promising strategy for combating localized infections and preventing their spread. Fungal infections pose a significant threat to public health, as conventional antifungal drugs often exhibit limited efficacy and systemic side effects. Copper nanoparticles have shown promise as novel antifungal agents, owing to their inherent biocidal properties. These nanoparticles can disrupt fungal cell membranes, interfere with vital cellular processes, and induce significant oxidative stress. Furthermore, copper nanoparticles exhibit broad-spectrum activity against various pathogenic fungi, including drug-resistant strains. The use of copper nanoparticles in antifungal therapy presents a viable alternative to conventional treatments and can help combat the rising incidence of fungal infections. Antimicrobial coatings play a crucial role in preventing the spread of infections in healthcare settings. Silica nanoparticles offer unique advantages in this context, as they can be incorporated into coatings, imparting antimicrobial properties to various surfaces. The high surface area of silica nanoparticles allows for increased contact with microbial pathogens, enhancing antimicrobial efficacy. Additionally, silica nanoparticles can be functionalized with antimicrobial agents or antimicrobial peptides, further augmenting their antimicrobial activity. The incorporation of silica nanoparticles into coatings holds immense potential for reducing the transmission of infections and improving hygiene practices. One of the challenges in antimicrobial therapy is delivering drugs to specific infection sites, thereby maximizing therapeutic efficacy while minimizing side effects. Magnetic nanoparticles, equipped with external magnetic fields, offer precise site-specific drug delivery. These nanoparticles can be guided to the target site using magnetic forces, increasing drug concentration at the infection site. Additionally, magnetic nanoparticles can be functionalized with antimicrobial agents, enabling synergistic antimicrobial effects. The use of magnetic nanoparticles in site-specific drug delivery represents a breakthrough in antimicrobial therapy, allowing for personalized and targeted treatment. Combining multiple nanoparticles with complementary properties has led to the development of hybrid nanoparticles, harnessing synergistic antimicrobial effects. These nanoparticles integrate the unique physicochemical characteristics of different materials, resulting in enhanced antimicrobial activity. For example, the combination of metallic and polymeric nanoparticles can leverage antimicrobial properties, sustained drug release, and targeted delivery capabilities. Hybrid nanoparticles hold great potential in overcoming microbial resistance and improving treatment outcomes, by combining the strengths of different nanoparticles in a single therapeutic entity. Photodynamic therapy, a non-invasive treatment modality that involves the use of photosensitizing agents and light, has gained attention in antimicrobial therapy. Nanoparticles can be utilized to enhance the delivery of photosensitizing agents, ensuring their precise localization within microbial cells. Upon exposure to specific wavelengths of light, these agents produce reactive oxygen species, causing microbial cell death. The use of nanoparticles in photodynamic therapy offers improved drug delivery and enhanced therapeutic outcomes, making it a promising approach for treating infections caused by drug-resistant pathogens. As nanotechnology progresses rapidly, the regulatory landscape surrounding nanoparticle-based antimicrobial products becomes increasingly complex. Ensuring the safety, efficacy, and quality of these products requires a comprehensive understanding of their physicochemical properties, toxicological profiles, and manufacturing processes. Regulators face challenges in establishing appropriate evaluation criteria and standardized testing methods for nanoparticle-based products. Moreover, the potential environmental impact of nanoparticle release and long-term effects on human health must be carefully addressed. Collaborative efforts between researchers, regulators, and industry stakeholders are vital in navigating these regulatory challenges and ensuring the responsible development and deployment of nanoparticle-based antimicrobial products. In conclusion, medical nanotechnology provides exciting opportunities in the field of antimicrobial therapy. The various types of nanoparticles, including metallic, polymeric, liposomal, and silicon nanoparticles, offer unique advantages in combating drug-resistant infections. Additionally, quantum dots, nanoemulsions, copper nanoparticles, magnetic nanoparticles, hybrid nanoparticles, and nanoparticle-mediated photodynamic therapy present novel strategies to address antimicrobial challenges. However, regulatory challenges must be carefully addressed to ensure the safe and effective use of nanoparticle-based antimicrobial products. With continued research and collaboration, medical nanotechnology holds immense potential in revolutionizing antimicrobial therapy and combating the alarming rise of drug-resistant infections.